In this lecture, we are going to introduce the very important protein involved in the action potential, and that's the sodium potassium pump, also referred to as the sodium potassium ATPase. This is a membrane bound protein that functions as a transporter, and this is found embedded in the membrane of neurons as well as muscle cells and other cells in the body. And the overall function of this enzyme is that it will chemically bind three sodium ions on the interior of the cell and move those ions to the exterior, and then take two potassium ions outside the cell and to move those into the cell. Now, the movements of both of these ions occur against the electrochemical gradients, as well as the concentration gradients for these two ions. And in order to move these ions, quote unquote, uphill, it does require the use of one ATP molecule for every 3 and 2 sodium and potassium ions, respectively, to move against their gradients. And the function in skeletal muscle cells, as well as neurons, is to establish and maintain the electrical potential of the cell under resting conditions. So this is an enzyme, a protein transporter that is active not only during the action potential, but is also active under resting conditions. Because the cell membrane of neurons and muscle cells do allow sodium to quote unquote leak into the cell, it's important to maintain the cell at its resting membrane potential of -60 by moving those 3 sodium ions out of the cell while bringing 2 potassium ions back in. And so in doing so, this sets up the energy state or the potential difference or the voltage. So the -60 is maintained by the action of the sodium potassium ATPase when the cell is not under activation conditions. Now, when the muscle cell or the neuron does generate an action potential, so when that membrane hits the threshold and then the cell will depolarize, then the sodium potassium ATPase becomes even more important in its role, because at this point it is attempting to restore the cell back to its ionic equilibrium state, which is the -60 millivolts on the interior of the cell. And so the sodium potassium ATPase, it is a protein functioning as an enzyme, and it's made up of 2 major subunits, what are just simply referred to as the alpha subunit and the beta subunit. So the alpha subunit is the catalytic and transport unit of the enzyme. So this is the portion of the overall enzyme that has binding sites for one ATP molecule, the cations in question here, sodium and potassium, and under experimental conditions, a compound referred to and pronounced as wabane is a specific inhibitor of the protein, and it will bind to the protein and not allow it to work. And this is a compound that's used in experimental conditions to examine sodium potassium ATPase physiology under a range of different conditions. The second smaller unit, or the beta subunit, functions to modulate the affinity of sodium and potassium to the actual protein. So by modulating the affinity, this portion of the protein will reduce the strength of the connection between these ions and the actual enzyme. And that's an advantage because when we want to extrude sodium from the interior of the cell towards the exterior, we want a very light, weak connection when that protein moves the sodium to the exterior of the cell, thereby allowing it to release. And so in turn, the beta subunit will help to maintain normal ATPase activity. This is just for visual purposes here. This is a representation, a graphical representation of the sodium potassium ATPase structure. And so as you see here, there are a number of transmembrane domains here that anchors the protein, the enzyme, into the membrane of the cell, pointing to the extracellular medium, and a rather extensive component that extends into the cytoplasm, because that's where the enzymes and that's where the ions and the ATP molecule reside. And so we have our much larger alpha subunit, our smaller beta subunit. And it's important to note that like many enzymes, the sodium potassium ATPase does exist as different isoforms, but that goes beyond the extent of this particular lecture. So in terms of the mechanism of how this works, there are what are considered 4 conformational states that relate to the shape of the enzyme itself, named E1, E1P, E2P, and E2 in a succession, as you can see here, where P refers to the domain, that portion of the enzyme where the ATP molecule binds. Specifically, the phosphoryl group of ATP will bind on the amino acid or the residue aspartate specifically. And the enzyme begins its function first by binding 3 sodium ions on the interior of the cell, as well as 1 ATP molecule. And that's just simply referred to as the E1 state. And that's really the start of this process. The second part is referred to as enzyme phosphorylation. And so ATP is useful when we spend it, which is to break up the phosphate bond holding the phosphate molecule onto the rest of the molecule. So ATP is hydrolyzed, and that forms a connection called phosphoaspartate. Remember that ATP binds to the aspartate residue, and then when the phosphate bond is broken, what that leaves behind is a phosphoaspartate part of that enzyme. Now, the stimulation for this step here is the presence of intracellular sodium. So if sodium has leaked into the cell in small numbers under resting conditions, that is a stimulant for the ATPase to begin its activity. Now, if sodium rushes into the cell because the cell has reached threshold, then that is a strong stimulus for ATPase activity. So the sodium potassium ATPase does modulate the rate of its conversions depending upon the magnitude of sodium in the cell. Number 3 here is called the E2P state. So that's our third step here. And the ion binding sites will evert or cause a change in the shape to where the binding sites will face the extracellular side of the cell. And in that state, in the E2P state, that change in the shape, the enzyme has a low affinity for sodium. And this is an advantage because we don't want sodium bound strongly to the enzyme when this part of the enzyme is facing the extracellular component of that cell. So it can easily release sodium into that extracellular space. So the E2 state, we see that sodium is released to the extracellular side. And then we can, at this point, we say that the enzyme is dephosphorylated. At that time, the phosphate group on the interior portion of the cell is released from the aspartate residue. And this puts the enzyme in a position to bind 2 potassium ions that are on the exterior part of the cell. And so in this state, the enzyme is considered unstable. once that phosphate group has been removed from the aspartate residue. And in doing so, the enzyme will seek its stable conformation and will evert back into the cytosol. In doing so, the affinity for potassium will decrease, thereby releasing potassium back into the cytoplasmic region of the cell. And the enzyme has now been restored back to its resting state, ready for the next cycle of moving sodium ions out of the cell and binding one ATP molecule to do that. So this ends this video on the introduction to the sodium-potassium ATP